So good afternoon, everyone. So here is the outline of my talk. I'm going to first present you what is the general knowledge about the gut microbiota. I will mention its development from birth, the influence of environmental factors, and then the consequence of its alteration in disease. So for an adult of 70 kilograms, you have approximately 2 kilograms of intestinal content. So of course, it's not only microbes, there is also a lot of water in our gut. We estimate that currently there are as many microorganisms in our gut than the number of cells in our body. Roughly 10 to the 13 microorganisms. And so for 1 gram of stool, there are more than 100 billions of microorganisms. In our cells, we have the human genome. And if we put together the genes present in the microbiome of one healthy subject, it represents 25 human genomes. In terms of size, showing that it's a huge mass of information. It's also quite difficult to handle this mass of information, and it has also a huge potential in terms of activity. So in the gut, we have bacteria. This is at least the part we know the best. And notably in the colon, most of these bacteria are strictly anaerobes belonging to these two major phyla, the Bacteroidetes and the Firmicutes. But we also know now that there are many other types of microorganisms in the gut, fungi, but also phages. Phages are viruses that target bacteria. So they are not targeting our human cells, but they are targeting bacteria. And so they can modulate the population of bacteria in the gut. There are other microorganisms such as archaea and also protists. So really a lot of microorganisms. So one of the issues when someone wants to study the gut microbiota is that only 30% of the dominant microbes are cultivable, at least easily. And so to study globally the gut microbiome, it is required to use culture-independent methods, and so basically to use sequencing technology. When you think about all these methods to study the gut microbiome, there is, of course, the possible study of the DNA. And this is what we call metagenomics, because we are not studying the genetic material of one organism, one bacteria, but the sum of the genetic information from several hundreds of microorganisms. It is also possible to study the information at the RNA level, and so in this case we call it metatranscriptomics. Possible to do it for the protein, and then it's metaproteomics. For the metabolites, the small molecules, so then it's metabolomics. And of course, at one point, we still have to come back to the bacteria and to grow them to understand what they are doing. So the gastrointestinal tract is not homogeneous in terms of environment, and so it's also not homogeneous in terms of microorganism. For example, you can see that the bacterial load increase from the upper GI to the lower GI tract. And in the same time, you see the oxygen concentration is decreasing from the proximal to the distal GI tract. And so the microorganisms are adapted to this change of environment, meaning that you don't have the same bacteria in the upper and in the lower GI tract. Another important aspect is that we have a microbiome which is specific to each subject. And so here, this is an old method used to characterize the microbiome. And here, each bar represents one bacteria. And you can see in these 14 healthy adults that the microbiome are different. Although some bacteria seems to be present in most of the subjects, you see some bars that are present in many people. And indeed, it has been shown that some bacteria are present in everyone. It is what we call the core microbiome. And there are probably around 15 to 20 bacteria in this case that you can find in everyone. The other important information is that the microorganisms in the gut do not organize themselves randomly. It's more like an ecosystem, so the microorganisms interact with each other and also with our human cells. And so there is a structured organization of the microorganism. It's not random. And so it has been shown in this paper, which is more than 10 years old now, that indeed there are three large types of organization of the microbiome in humans that has been called enterotypes. And indeed, this result has been reproduced in other studies on humans, but also in other animals, such as chimpanzee, for example. And so to better understand this concept, you have to know a little bit about ecology. And for this, we usually use this type of picture. So you can imagine that the microbiome, your microbiome, is this balls, which is in this valley on this geographic zone. And you can see that it's at the bottom of the valley, meaning that if you have a disturbance of this microbiome, the ball will move, but then it will come back to the initial state, and this is what we call the resilience. However, if the perturbation is too strong, then the balls will change of valley and will go into another one, and this is what we call an alternative stable state, and this event is called a critical transition. And however, this geographic profile is not fixed. It's not. It can move, and indeed, based on different factors, you can think about moving this geographic profile. And when you are applying this concept to microbiome and health and disease, you can easily understand that the first stable state is the health status, and the alternative stable state is a disease status. And you can move this this geographic profile with diet or drugs. So as I mentioned, we don't have the same microbiome. This is data on roughly 15 healthy subjects, and you can see that their composition of microbiome is very different from one to the other. So when we are talking about composition, we are talking about the name of the bacteria, basically. And however, the name of the bacteria, it's not so interesting. What is interesting is what the bacteria can do. And this is what we call the functions. And when you look at the functions of the microbiome in these same 15 subjects, you can see that globally, these functions are very stable and conserved from one subject to another, which is actually not so surprising and even reassuring to know that the function in the gut microbiome of an healthy subject is roughly the same as the function present in another subject living at least in the same environment. So when we are talking about function of the microbiome, we are thinking about the function that the microbiome is doing for us, for the human cells. And there are many, many different functions. So I will not mention all of them, but just give you some big examples. So first, the microbiome has metabolic functions. It controls intestinal epithelial cells differentiation and proliferation. It metabolizes xenobiotics. It's also involved in the synthesis of vitamins, such as vitamin K, for example. It also ferments non-digestive dietary residues. This is the case with fibers that you can find in fruits and vegetables, and these fibers are non-digestible by our human enzyme. And this is the microbiome in the colon that is able to digest these fibers for us. It will take what it needs from it, of course, but then it will also produce short-chain fatty acid from the digestion of these fibers. And these short-chain fatty acids are indeed very important molecules for intestinal health and more globally for the human health. The microbiome is also involved in maintaining the barrier function. So, of course, the main function of the gut is to absorb calories, but in the same time, it should not be completely open to the external world. So there is a kind of protection against it, and this is what we call the barrier function. And the microbiome is involved in this barrier function, notably by reinforcing the tight junction between the epithelial cells. So basically the gut is not leaky. It also induces the secretion of immunoglobulin A in the lumen. It also induces the production of antimicrobial peptides by our cells. And so these peptides are basically natural antibiotics that the human cells are producing. It also plays a major role in the immunity by stimulating and maturating the intestinal immune system and even globally the global immune system. It is protecting us against infection, particularly intestinal infection, by competing with pathogens for nutrients and receptors and by producing also antimicrobial factors. And finally, it can also act everywhere in the gut, including in the brain. So what is particularly interesting with the gut microbiota is that it is impacted by both our cells, so genetics, immunity, but also by environmental factors. And when we are discussing environmental factors with the gut microbiome, there is indeed two different periods in life. First, you have everything happening early in life, and this can have an effect on the first setup of the microbiome in our gut, because as you probably know, in the maternal uterus, we are sterile. There is no living microorganism in the maternal uterus, and so we basically meet the microbial world after birth. And based on what is happening at this time, there can be effect on the microbiome, and many factors can be at play, such as the mode of delivery, C-section, or vaginal delivery, the types of diet, maternal feeding or artificial feeding, the place you live. Of course, in a microbial point of view, it's really not the same to live in the middle of a big city or in the countryside, and other factors such as exposition to antibiotics. And we think now that the first host So we think that the first host microbes interactions occurring early in life are very important and can indeed impact the immune system and even globally our physiology, and if these interactions are altered, it can lead to a miseducation of the immune system and potentially to the development of immune-related disease later in life. Actually, this has been demonstrated, at least in mice, in this study published 3 years ago by colleagues from the Pasteur Institute. It was shown that perturbation of the microbiota at the weaning period using antibiotics will lead to a miseducation of the immune system and to the increased susceptibility to intestinal inflammation later in life. So of course, not everything is done during the first day of life, and even during adulthood, many factors can impact the gut microbiome. The first very important factor is diet, and notably the richness in fibers that I mentioned earlier. So the richness in fruits and vegetables are very important for the good shape of the microbiome, while having this type of diet is bad because it's low in fibers and high in bad component for the microbiome. Ultra transformed diet is also bad, notably emulsifiers, but also conservative and so on. Now it is known to be detrimental for the microbiome. Of course, exposition to antibiotics is also bad, although obviously antibiotics are very important drugs, but the only thing is that it should be used only when it's necessary and only in this case. Now we have more and more data showing that other drugs besides antibiotics can impact the gut microbiome, and then other factors such as smoking also can have impact. And if these factors are at play, they can perturbate the gut microbiota, alter its composition and function, and this can have consequences on host. So let's move to the second aspect, which is the development from birth. It will be much shorter. So here, I mean, as I mentioned earlier, at birth, we are sterile. There is no living microorganism in the maternal uterus. And then when at birth, there is a progressive colonization of our gut by different waves of colonizing microorganisms. And so first you have notably bacteria that are able to live in presence of oxygen. And then these bacteria in the gut will consume the oxygen so that bacteria living in anaerobic condition will be able to colonize. And so what is interesting is that the immune system basically develops in parallel and under the direct influence of the gut microbiota colonization process during the early days. So I already mentioned it, but many factors can shape the neonatal microbiome. I mentioned first, what is interesting is that even before birth, there is some impact of the maternal microbiome on the babies because molecules produced by the maternal microbiome can go through the blood and reach the baby and have an effect during gestation. Then, of course, there is birth, and I mentioned vaginal versus cesarean delivery. And then there is the milk consumption, solid food introduction, and this is all the factors I mentioned. And then there is basically the adult microbiome that is set up roughly between 3 and 5 years old, at least according to the last studies. And so if we take an example here about the effect of the mode of delivery, here you can see the microbiome composition of children born by vaginal delivery and here by C-section. And you can see here, this is the microbiome at 1 year, at 12 months. You can see that globally between 12 and 36 months, there is no major differences between children born by vaginal birth or C-section. However, if you look earlier, so during the first year of life, you see that there is indeed major differences, and which is basically the proof that there is an impact of the mode of delivery. And of course, as I mentioned earlier, we think that these first months of life are very important because they probably lead to an imprinting of the immune system and globally of our human physiology, and suggesting that it could have potential long-term effect, including during adulthood. So what about environmental factors? I will give you a few examples of the influence of environmental factors. So let's take, for example, antibiotics. You can imagine that this is a kind of environmental factor for the microbiome. So here, I will ask you to focus only on this blue line. And this figure represents the diversity of the microbiome, meaning the number of different species present in the gut. And here on the x-axis, this is the time. This gray bar represents the exposition to an antibiotics. And so what you see is that here, this blue line remains to globally a relatively high diversity. But when the patient receives antibiotics, you see the diversity decreasing very drastically to a very low level. And when you stop the antibiotics, the diversity comes back to the level which is indeed quite close of the initial diversity. And this is, again, the resilience features that I mentioned earlier. However, in this case, if you repeat the perturbation with another course of antibiotics, you see that the diversity is decreasing again, but then it does not come back to the initial state, and you have a kind of decreased diversity which can remain stable, and this is this alternative stable state that I mentioned earlier. This I will skip. So what about diet? So I mentioned diet is a very important environmental factor for the microbiome. So this is an experiment performed in mice. Mice are eating usually mostly vegetable-based diet. And so here, in this case, this is basically a measurement of the microbiome. And you can see that when you change the diet using an animal-based diet, you see that the microbiome is changing, and it's going very fast. You see here, this is in 2, 3, 4 days, you have a major change in the microbiome and when you come back to the initial diet, then again in a few days it come back to the initial state, clearly showing a very dramatic and very rapid effect of diet on the microbiome. And this is also now known in humans. So this is a very famous study published almost 15 years ago now, comparing the microbiome of children living in rural Africa, so in Burkina Faso, and in Europe, actually it was in Italy. You can see that the microbiome of these two groups of children are very different. And actually, what you can notably see here is that these green bacteria are highly represented in children from Burkina Faso, but they are not seen at all in children from Europe. And these bacteria, particularly the xylanobacter here, are able to digest very strong fibers present in millet and sorghum, which is a very important source of energy for this population. So basically, the microbiome here is adapted to the diet habits of this population, and it helps digest these fibers and get energy from it. While in Europe, unfortunately, there are more and more this type of diet with very low fibers, and so these bacteria who are eating fibers and who are producing these good molecules I mentioned earlier, the short-chain fatty acid, are decreasing drastically. And this is very important because this decrease in diet may have some long-term consequences and even transgenerational consequences. So this is a very nice experiment in mice. So in this experiment, the mice were fed with a diet either low in fibers or high in fibers. And what you can see here, so in green, this is the mice fed a high fiber diet, and you can see that during the time, so here this is in week, globally the microbiome does not change. When you fed the mice with a low fiber diet for a few weeks, you see the change, a massive change in the microbiome. And then when you come back to the high fiber diet, you can see that the microbiome tend to go back to the initial state. However, the question now is to know what is happening when you follow this perturbation of the microbiome with low fiber diet for several generations. Can you still go back to the initial state? And you are going to see the response, the answer to that. So here is exactly what we saw before. This is the first generation. So when you do this for one generation and then you come back to the initial state, to the initial diet, you can see that globally you tend to go back to the normal microbiota. However, if you do the perturbation of the microbiome with low fiber diet for 2, 3, or 4 generations, even if you change and you come back later with a high fiber diet, you cannot go back to the initial microbiome. And actually, this is quite logical, because if you completely get rid of the bacteria that are able to use these fibers, even if you change the diet, the bacteria are not going to come back. And this is particularly important because this Western diet now, we have it for a long time, and it is possible that we have this type of transgenerational effect regarding the microbiome, even in humans, actually. So in this experiment, the only possibility to restore the normal microbiome was indeed to perform fecal microbiota transplantation, so to bring back really new microbes into the system. So now let's move to disease. If this microbiome is so important for health, of course, you can imagine that when it is altered, it can play a role in disease. And now there are many proof of this implication of the microbiome in obesity, atherosclerosis, in inflammatory disease such as inflammatory bowel disease, arthritis, and even in brain conditions. Just going to give you an example based on one type of disease, which is actually my specialty, inflammatory bowel disease. So so maybe just one word about this disease. So there are two diseases, Crohn's disease and ulcerative colitis. This disease affects a lot of people. So there are several millions of patients in Europe and as many in the US. They are affecting relatively young subjects, altering their quality of life. And we don't have a curative treatment for this disease. We have suspensive treatment, and the aim currently is to try to block the overactivation of the immune system in the gut, which is present in this condition. And of course, as it is in the gut, and there is inflammation in this disease, the microbiome was suspected for a long time. For a long time, it was only suspected, but now we can clearly say that the microbiome does play a role in the pathogenesis. Why? So there are many reasons. First, if we modify the microbiome in mice model of intestinal inflammation, or in humans also using fecal microbiota transplantation, and I'm going to show you some example, indeed, in this setting, there is an effect on the disease. And another very important argument is that the fecal microbiota of patients with IBD indeed exhibit pro-inflammatory effect. If you take it and transfer it to germ-free mice, the mice will be more susceptible to colitis compared to mice colonized with a normal microbiome. So in this disease, patients have indeed an abnormal microbiota. On this type of figure, each dot represents the microbiome of one subject, and if two dots are close, it means that the microbiome are similar. If the dots are far, it means that they are different. And here you can clearly see that the healthy subjects are here in blue, and the patient with IBD with active flare are here in red, while the patient with IBD in remission are basically overlapping between the two groups. So the microbiome is different. Another characteristics of this microbiome is that its diversity is decreased compared to healthy subjects. You can see that the diversity of the microbiome is decreased. And then in terms of composition, what we can see is that in healthy subjects, you can see these two dominant bacterial groups, the Firmicutes in blue and the Bacteroidetes in green. And here what you can see in patients with IBD is that the proportion of Firmicutes decreased, and you can see a new group appearing, which are the proteobacteria. And this group is here in orange. And so we showed already a few years ago now that among the bacteria that are decreased in IBD, there is this bacteria, Faecalibacterium prusnitzii, and these bacteria exhibit anti-inflammatory effect in many settings, in colitis model, in human cells also. And it's not written here, but there is also a role for some bacteria present in this orange group, notably for Escherichia coli, which exhibit pro-inflammatory effect. So you have two things in parallel. You have decrease of anti-inflammatory bacteria, and you have increase in pro-inflammatory bacteria. So this I'm going to skip. Okay. So maybe just one word about potential use of the microbiome as treatment. So you probably know about probiotics, conventional probiotics, which are mostly obtained from lactic acid bacteria and bifidobacteria from fermented milk products. These bacteria are traditionally used in food. So these are conventional probiotics. But now there is a development of a new class of probiotics called next generation probiotics or live biotherapeutics products. And these microorganisms, instead of coming from dietary sources, they are isolated from the gut microbiota itself and selected for biological effect, rationally selected. So, of course, when you think of a bacteria, of an intestinal bacteria, it's probably more efficient to have effect on health than a bacteria isolated from a cheese, for example. So here is one example of this next generation probiotics. I mentioned earlier this bacteria, Faecalibacterium prusnitzii. This is indeed one of the bacteria belonging to the core microbiome. So these 15 to 20 bacteria that are present in everyone. And we showed that this bacteria and its abundance is indeed decreased in patients with inflammatory bowel disease. And this was shown now by many groups around the world. We also showed that the level of this bacteria indeed is able to predict the risk of relapse. So the lower amount of this bacteria you have, the higher is your risk of relapse, for example, in the context of Crohn's disease. We also showed that these bacteria indeed exhibit anti-inflammatory effect, both in vitro, in different models, and also in colitis model in mice. So we think that this bacteria could be a new drug, and indeed there is a clinical trial currently evaluating its effect in Crohn's disease. So this is the concept of next generation probiotics. Then a much drastic way to modify the microbiome is fecal microbiota transplantation. So the concept is indeed very simple. The idea is to replace an altered microbiome for which you think it plays a role in the disease by an healthy one, with, of course, the hope to have a positive effect. So currently, there is only one indication of this treatment in routine clinical practice, and this indication is an infectious disease, a gastrointestinal infectious disease caused by a bacteria called Clostridioides difficile. And in this setting, fecal microbiota transplantation are highly effective, more than 90% of success compared to usual antibiotics such as vancomycin. And so this treatment was indeed considered to be very useful in this indication, and everywhere in the world, in Europe, in US, it was added into all the different guidelines for the treatment of this infection by Clostridioides difficile. In other disease, of course, for which we think the microbiome is involved, it is very attractive to test fecal microbiota transplantation. And indeed, it has been done in inflammatory bowel disease and notably in ulcerative colitis. And in all these studies, the idea was to try to treat the patient with fecal microbiota transplantation. And globally, these are the results of the first 4 studies that evaluated FMT in ulcerative colitis. Globally, the effect were positive, but one of the problems was, I mean, there was indeed several questions. First question was related to the small size of the study. The other aspect is that there was a high heterogeneity in terms of procedure. And then, of course, the result we got here were very different from the result I showed you for Clostridioides difficile. To remember, for Clostridioides difficile, the efficacy was more than 90%. Here, the remission rate with fecal microbiota transplantation is between 25 and 35%. So it's clearly not as effective as in Clostridioides difficile infection. We did ourselves a study in Crohn's disease with another strategy. So here, instead of treating the inflammation with FMT, first we induced the remission with an immunosuppressant, which are corticosteroids. And when the patient achieved remission, then he received either one fecal microbiota transplantation through a colonoscopy or a sham transplantation, and then the corticosteroids were tapered for several weeks. And basically, this is what we showed. So we observed all the patients were in clinical remission at the randomization because it was mandatory to enter the study. And then you can see that patients in the FMT arm have a lower relapse rate compared to the patient in the control arm in blue. And if you look at other parameters, so here this is the endoscopic index of severity, you can see that in the FMT arm, after 6 weeks after FMT, the inflammation is decreased, while in the sham transplantation group, in the control group, there is no change in the intestinal inflammation. And this is basically same picture when you look at a marker of systemic inflammation. In the FMT arm, here you can see there is indeed 5 patients here, and there is no change in this inflammation marker between the time of transplantation and 6 weeks later. While in the sham transplantation group, you can see that already at 6 weeks, this marker is increasing, suggesting that the disease is already less and less controlled. So, in conclusion, the gut microbiota is indeed composed of trillions of microorganisms, mainly bacteria. At least this is the part we know the best. And it is under the double influence of genetics and environmental factors. It's a key factor for important physiological functions, including metabolism, immunity, and brain function. And an altered gut microbiota can play a role in intestinal and also in extra-intestinal disease. And of course, this is a promising tool and target as a biomarker, but also treatment. However, we have to remind that microbiota is only one actor in health, because if you read newspaper nowadays, you can feel sometimes that the microbiota is responsible for everything. But indeed, for many diseases, we don't know what is exactly the weight of the microbiome. So in some diseases, the microbiome is the main actor. And this is the case, notably with the infection I mentioned, the infection by Clostridioides difficile bacteria. In IBD, we also know that the microbiome is an important actor, but there is the role of other factors, obviously. And then for many other diseases, we think the microbiome is involved, and we have even proof of that, but we don't know what is the weight of the microbiome. So maybe the microbiome is involved, but it's not a major actor. It might be only a minor actor. So this is also a very important field of research to understand which disease are really impacted by the microbiome. In terms of treatment, of course, I mentioned fecal microbiota transplantation. It's a very attractive treatment, but it's also very complex. We clearly do not know exactly what we are giving to the patient. We are not able to define the composition of the FMT. So probably to treat patients with chronic and very, very frequent disease, such as IBD, but also diabetes and other, we'll have to move to more simple treatment. Can be a mix of defined bacteria. This is called consortium, or even single bacteria. And all this fall into the concept of the next generation probiotics I mentioned with this example of Faecalibacterium progeny. And if we well understand the mechanism underlying this effect, obviously this is happening through molecules either present on the bacterial wall or produced by the bacteria. And if we can identify this molecule, we can also use it directly as drug. And this is the concept of postbiotics. I did not mention it too much, but there are now more and more data showing that the microbiome is likely responsible, at least partly responsible, of the heterogeneity in drug response and toxicity. It should be taken into account in drug development for pharmacokinetics and pharmacodynamics. And as I mentioned, it's already identified as a source of new treatment. Thank you very much, and I'm happy to answer any question you have.
Yes, thank you very much. We have some time now for discussion, so if anyone has questions, ask. And if not, I wanted to ask, what impact does gender have on the gut microbiome, if it has any?
So I would say at baseline, I mean, in usual conditions, there is no major effect of gender. However, there is effect, for example, during pregnancy. So we see that the microbiome changed during pregnancy, very likely because of hormones. And so in the same woman, the microbiome is not the same at the first trimester and at the last trimester. But we don't know yet how this is working.
Okay, thank you. And then the gut microbiome, it goes back to the initial one after pregnancy?
Yes, yes.
Yeah. Okay, thank you. Does anyone else have any questions? You can just unmute yourself and ask.
Hello, can you hear me, please?
Yes.
Yeah. First of all, thank you for amazing lecture. And I know it's not exactly your specialty, but what class of antibiotics is or has the least impact on gut microbiota?
Yes,
if you can tell it.
Sure. So you are right. Not all antibiotics have same effect on the microbiome, obviously. So I can tell you what are the antibiotics that have the strongest impact first. And these are the antibiotics acting on anaerobes. And so notably all the antibiotics such as amoxicillin acid clavulanic. So bacteria targeting anaerobes are very damaging on the microbiome. And then you have other antibiotics that are have less effect. This is the case, for example, for macrolide or for Bactrim. These antibiotics have less effect. Even tetracycline, they have less effect on the microbiome.
Thank you very much.
Are there any other questions?
Hello, I would like to ask how exactly the food additives or the food emulsifiers are bad for the gut.
Yeah.
Thank you.
So in the gut, you have on the epithelial cells, you have a very important layer of mucus. And this mucus is a protective layer for the gut, but it's also important for the ecosystem. And indeed, it has been shown that emulsifiers are very bad for this mucus layer, and it basically decreases the thickness of this mucus layer. And this has been shown in animals, and it's increase the susceptibility to many settings in animals, such as intestinal inflammation, but also metabolic disorders, and so on. And there are also large epidemiological studies showing that the more ultra-transformed food you eat, the higher is your risk of IBD.
Okay, thank you.
Okay, so we still have some time for questions. So if anyone has a question. Maybe I wanted to ask one again. The fecal microbiota transplantation, is it from random donors, or are there like...
Sure, no, it's not from random donors, of course. We screen donors regarding safety mostly. So first, we want donors that have a good microbiome. So we exclude donors who received antibiotics recently. We exclude donors who have any chronic disease, who have traveled into exotic place. And then we do a very large screen for pathogens in blood, in stools, for also antibiotics resistant bacteria. So it's very, very large. And so globally, less than 3% of the candidates are indeed able to donate. So it's very, very difficult.
Mm-hmm. And is there also a correlation with the family member, or it doesn't matter?
So for FMT, no, we don't. I mean, at one point in the early days, we were using family members, but now we try to avoid it because what we try to have is now kind of bank with different stool preparation already available. And so if it's needed, we can use it immediately. Otherwise, if we have to wait for the family member, it can be very complex. And it's also a big problem when you find something in the family member. So you can imagine you have a man with a Clostridioides difficile. You want to do an FMT. You get the stool from his wife, and then you realize the wife has a disease like sexually transmitted or something like that. So it can be very can be associated with some issues. So we try to avoid.
Okay, thank you. Okay, well, last chance for questions.
Okay, good evening. I wonder if artificial sweeteners have some effect on the gut microbiome.
Yes, yeah, very, very good question. Yes, it does. They have effect because these sweeteners, they are not, actually it's like fibers. They are not degraded by our human enzyme, but they are degraded by the bacteria. And indeed, I don't know if you tried to eat a whole box of this kind of artificial sweetener. you will probably get diarrhea because this will impact your microbiome and also attract water in the gut. And yes, so definitely it has effect.
Thank you.
Okay, then if there are not any more questions, thank you very much for the lecture. It's very interesting, and thank you so much. Thank you, students, for coming.
Thank you.
And maybe see you in the future again.
Sure. Bye-bye.
Bye.